This episode of our series covers the work of Gregor Mendel on genetics, why his ideas weren't popular at the time, and further developments in genetics after him. Let's begin with Gregor Mendel, an Austrian monk whose work in the 1860s laid the foundation for modern genetics. He carried out a series of breeding experiments using pea plants, selecting them because they had clear, contrasting features. What made Mendel's approach special was that he carefully tracked specific traits such as plant height, flower color, and seed shape through multiple generations. Think about it. He literally counted thousands of individual pea plants and precisely recorded their characteristics. Through these painstaking experiments, Mendel observed that traits weren't simply blended together in offspring as was commonly believed at the time. Instead, traits appeared to be passed down as discrete units that remained unchanged from generation to generation. Mendel called these units factors, what we now know as genes. He concluded that each trait was determined by two factors, one inherited from each parent, and that some factors were dominant over others. For example, when he crossed tall pea plants with short ones, the offspring weren't medium height as people expected. Instead, they were all tall, showing that the tall factor was dominant over the short factor. Despite publishing his findings in 1866, Mendel's work remained largely unrecognized during his lifetime. But why was such groundbreaking research ignored? There were several reasons for this scientific oversight. First, Mendel published his work in an obscure scientific journal that wasn't widely read by the international scientific community. Second, his statistical approach to biology was unusual for the time. Most biologists weren't mathematically trained and struggled to understand his numerical analysis. The idea of using maths to study living things was quite revolutionary. Perhaps most importantly, the scientific community simply wasn't ready for Mendel's ideas. His theories contradicted the prevailing belief that inherited traits were blended together, like mixing paint colors. There was also no understanding of the physical basis for his mysterious factors. Scientists couldn't see or touch them. It wasn't until the early 20th century that the significance of his work was finally recognized. The rediscovery of Mendel's research coincided perfectly with new observations about cell biology. In the late 19th century, scientists had developed much better microscopes that allowed them to observe chromosomes during cell division. They could see these thread-like structures moving around inside cells, but didn't yet understand what they did. This was like having a key but not knowing what door it unlocked. In the early 20th century, scientists began to notice something remarkable. Chromosomes and Mendel's factors behaved in remarkably similar ways during reproduction. Both seemed to be passed from parent to offspring in predictable patterns. This led to the crucial insight that genes might actually be located on chromosomes. This connection between Mendel's abstract factors and the physical structures inside cells was a major breakthrough in scientific understanding. Scientists now had a mechanism to explain how true traits could be inherited. The chromosomes were like tiny filing cabinets carrying genetic information. But the story doesn't end there. In the mid 20th century, another huge advance came with the discovery of DNA structure by Watson and Crick in 1953. This built on the work of many other scientists, including Rosalind Franklin, whose X-ray crystallography was crucial to understanding DNA's shape. Once the double helix structure of DNA was understood, scientists could begin to work out exactly how genes function at the molecular level. They discovered that genes contain the instructions for making proteins, which carry out most of the important functions in our cells. All of this scientific work by many different scientists over more than a century led to the development of what we now call gene theory. This is our modern understanding of how characteristics are inherited through genes and how they function in our bodies. This story of the development of genetic understanding is a perfect example of how science progresses. Ideas are proposed, tested, sometimes forgotten, then rediscovered and refined as technology and knowledge advance. Scientific understanding rarely comes from a single breakthrough, but develops gradually through the collective work of many scientists over time. Each discovery builds upon previous work, creating an ever growing tower of knowledge. Our next episode will explore the evidence for evolution, including fossils and antibiotic resistance in bacteria.